All right. Thank you, everybody, for coming to our Rosmond Evening Talk. It's great to see everybody here. I'm very proud of our interns this year, so I'm very excited for them talk a lot about their summer. We have coffee and refreshments over here. The bathrooms are down the hall right here. I'd like to give a big thank you to Bernard and Courtney for all their support throughout the season, as well as Derek and Renee and Jordan, all of which supported us throughout the season. And also a big thank you to the Rosmond family. The Rosmond Evening Talk is fully funded by the Rosmond Family Education Fund, which is an endowment that honors the Fred Rosmond and his family and their contributions to the forest community as well as forestry as a whole. So without further ado, our interns. Our fearless crew leader, Nina Perry. I'd also like to extend a huge thank you to everyone here for taking an hour out of your Tuesday to learn about our experience this summer and the projects that we've been working on. My name is Sophia. My name is Anna. My name is Bella.
My name is Dave.
I'm Stella. And today we will be discussing T3 PVC and plant ID. Please look to the next slide. You'll see that we've had a very busy summer. Primarily, we have worked on plot implementation on a project that we'll be discussing at length in a moment. We had some fun educational field trips, and for the first couple of weeks, we worked on an ethnoforestry project, which Nate will be discussing now.
So, as Sophia mentioned, first project we worked on was ethnoforestry, which is located on a small, about 5-acre experimental plot out on the way to a bush. So ethnoforestry is a people-focused, community-based forestry practice, which is aimed at creating healthy, early sterile understory habitat, as well as creating understory plants that are desirable for foraging, either socially or economically in the community. Ethnoforestry was developed over many rounds of community input sessions, mostly through focus groups here at ONRC. And they were asked the question, what would you want to see in your working forest, or what do you miss in forests? A lot of the research and findings at ethnoforestry are to be scaled up and used in larger scale treatments at operational scales, such as in T3, and also can be directly taken to modern management forestry practices. So our role within ethnoforestry was that of data collection. We collected data on how the trees were growing under a multitude of different forestry treatments, as well as natural and planted regrowth composition. And importantly, biomass data was also collected from each plot. Ethnoforestry is set up with a number of different units. Each unit represents one of these treatments, and there are about 6 plots per unit. We would go in and characterize each of these units with each of the plants that we saw in there. What is the percent cover of each of those plants? We would also say what the ground cover was, whether that be wood, whether that be rock or their soil, moss, and one of our favorites, basal rosette, which is basically leaf of plants lying flat on the ground, becoming part of the surface. That is very characteristic of this guy here, Hyra, which is the USDA code for Ericaceae, which was everywhere in ethnoforestry. Sophia's allergies are in that for sure. Each plot, we were also able to take geospatial data and tree LiDAR visibility data. For the tree data, we would do productivity analysis by measuring each of the saplings, taking its circumference, LiDAR visibility helps later when they flow LiDAR over the plot. And they kind of know what they're looking for. If we visually say, yes, this could be seen by LiDAR, or no, it's obstructed either by other saplings or other trees or plants nearby. And we got to have some pretty cool insight into how they do these remote sensing missions by flying one of these multispectral imagery drones. Shout out Miles, who let us fly this drone around, gave us a pretty good insight to how you can do remote sensing on large scale forestry experiments, and also a lot of other aspects of natural science. So Plant ID, a very fun and exciting and fulfilling challenge that we all underwent. A lot of us have taken ESRM 201, which is one of the UW prerequisite classes for environmental science, which is very heavy on the plant ID. But when we got out in the field, there are a lot of species that we saw out there that we do not cover in those classes. Most of them, in fact, and in that case, this book, Pochar McKinnon, Plants of the Northwest Coast, was immensely helpful. There's a lot of nuance between different species, like these two species of saxifrage. A lot of times it's just a small difference in leaf shape or the color of the stem or the flower that can give you the difference. And we like to be precise, so we will painstakingly go through the book for a long time to find the right species. Graminoids were especially tough, which are grasses, sedges, and rushes, which cover a lot of plot a lot of the time. And if we were stumped, we always could phone a friend, or through Nina, and also Dr. Courtney Bobson. She was with us, and they were very, very helpful. Each of the plots at UW at ethnoforestry went through a series of either one of 9 different treatments. And so the treatments included a type of wildlife deterrent, either a physical boundary in a fence, or each of the units were completely applied with an herbivore repellent, which was made entirely of possum blood. It worked well. This was crossed with differing understory treatments. The first one was agroforestry, in which understory species, these, which were some of the species that were brought up by community members, things they would like to see in their forests. They were planted in 0.5 meter spacing in rows away from each other. And these were planted alongside 180 trees per acre of Douglas fir. Early sterile management is a similar condition with the same understory species planted in rows 2 meters away. But this also included the addition of red alder. So we could explore the effects of its very efficient nitrogen fixability on the development of these Douglas fir seedlings, and also the understory. And there's also the control, which was only Douglas fir, but slightly denser, about 300 trees per acre. So how do we do it all? Plants of the Pacific Northwest Coast Identification Guide helped out a lot, of course, as I said. We also were able to use each of these plots. We would find these set rebar in the ground because each year they go back to the same plot and monitor how they change. And we would use 1.5 meter PVC pipes to set up an area, a square, which we can characterize what is in that square. We would also use the Jabot and Haglov and transponder for any geospatial data that we would need. This would usually look like setting up a Jabot, which is just a very highly accurate GPS somewhere in the middle of the plot, put the transponder somewhere close, and you use the Haglop and the transponder together. The Haglop can get the exact distance and the exact azimuth away from where the transponder is. So if you set the measurement from the Jabot to the transponder, you get what that distance and what that azimuth is. And then you can take the same measurements from anywhere in the plot to the same transponder and later georeference any of the trees or plots to LiDAR maps that could use the Javad data to find. We would also use the BioCube booklet, which I will get into in a sec, and data sheets to report all of the findings. So what is a BioCube? BioCube is basically used as a visual representation of any biomass in any plot that we come across. We obtain this information by first finding a 3x3 meter space that would be representative of a lot of the rest of the plot. And then we set up this square like so, and cut it all down with our trusty siblings. I would not want to be a fox though in a BioCube plot. We then collect all of the biomass, bag it, take it back to ONRC, weigh that. Then this biomass gets dried in the drying oven for 48 hours, and dry weight can be taken after that. These two measurements can then be used to calculate total biomass per hectare, which is the measurement we use to figure out how much biomass is in a plot. We take all of the pictures that we have of these biocubes and have a booklet that we can flip through, and we find the closest representative picture to what we're looking at. And then we can also visually add or subtract any percentage as we see fit for the most part. Another really interesting thing about this biomass information is it can be used to estimate biomass across very large scales. Stacy Dixon, who works in the Edel lab at UW, is doing this at ethnoforestry in using all of our biomass information that we've collected, along with the geospatial information using our Javad and Haglöf. She overlays that onto LiDAR and multispectral imagery produced maps, covering them, and then interpolates the space in between to find out what the biomass is across this 5 acres. And this process can also be scaled up to larger scale experiments like the T3 Watershed Experiment.
So let's zoom out a little bit, and I'm going to talk about the T3 Watershed Experiment, which is a version of the scaled up experiments that make us referring to. So the T3 Watershed Experiment is a large scale, long-term experiment that was designed with one question in mind. How can we manage our forests so that they work for both people and nature, especially as the climate changes? So this experiment took place in the Olympic Experimental State Forest on about 20,000 acres of public land here on Washington's Olympic Peninsula. What's different about this study is that rather than doing small test plots tucked away in research areas, we're working at an operational scale right alongside logging crews and foresters and land managers. And what this means is that if something works well here, it has the potential to be implemented in other working forests in the region. So this experiment has brought together scientists, foresters, and most importantly, the voices of the people who live, work, and have deep cultural ties to these lands. As they mentioned from the very beginning, this project has involved local community members and tribes in forming the ideas that are now being tested. Our overall goal is to expand the toolbox for forest management so that we have more than just 1 or 2 standard approaches. We want options so that we can balance timber production with ecological health, and so that we can protect wildlife and respect cultural traditions. In short, we're preparing for an uncertain climate future by testing these solutions now. So the T3 Watershed Experiment is testing a very large range of these solutions. And this summer, the crew and I were able to work on and learn about a few of the aspects of this project. but we spent most of our time, as you saw from the calendar, on the EVDP plot implementation. EVDP, ethnoforestry, variable density planting project. We're gonna dissect that, don't you worry. Next slide. So, as Nate mentioned, ethnoforestry is people-based forest management practices. The VDP, variable density planting, all that means is that rather than planting trees evenly spaced across the land, we're planting them in clumps and leaving open spaces in between. Now, the idea behind EVDP came directly from feedback that ONRC received from community members and tribes from the Olympic Peninsula that were concerned about the decline of deer and elk populations, and they were also concerned about the loss of understory species that are important culturally, economically, personally, salal, berries, medicinal herbs. There's so much important stuff. And the particular sites chosen for the EVDP experiment are on particularly rugged, steep slopes. And that means that while we had just the most wonderful time setting up the plots on these sites, it is actually difficult for humans to go in and harvest there. So rather than focusing on human foraging for this experiment, we're focusing on wildlife. So the big question is, how can we, or is it possible to balance, to have a productive forest with timber while also providing wildlife with rich habitat and food? So now let's discuss the differences between standard practices and EVDP. Standard practice generally is after the timber is harvested, all of the trees are replanted within 2 years. And before replanting, herbicide is used in order to reduce competition. And when they do replant, they replant the trees densely and mostly with Douglas fir. In the first couple of years before the herbicide treatments, you'll get a flush of wildflowers and grasses. But as the trees grow closer together, the canopy closes and sunlight isn't able to penetrate to the forest floor. And then you lose most of the remaining understory. Now, that is very bad news for wildlife because they depend on these early seral conditions. Early seral meaning the open, sunny stage of forest growth where food is very abundant for wildlife. And now let's talk about EVDP. EVDP is planting trees in clusters after harvesting, as opposed to planting them evenly across the landscape. And these clusters leave open spaces where sunlight can reach the forest floor, and a wider variety of plants are able to grow for a longer period of time. And this is really important for our life. And the things that we're going to be studying are the effects on tree growth, including crown development, wind throw risk, which is, are the trees gonna knock down? How are they gonna respond to the wind? And we're also going to be observing the different plants and animals that are going to be coming into these open spaces. We expect to see more we expect to see more wildflowers and shrubs, which will provide food for our elk, our deer, our pollinators, and many more wildlife.
So let's talk about the prescriptions. The prescriptions are unique, different numbers of trees per clump, and then the distance between each tree. And then within the EVDP project, there we have 4 different locations, which are A through D block. This is a diagram of A block. And then, so we have 5 different spacing prescriptions plus a control for each block like this one. And this is consistent across the blocks. And each one is listed here. So we have the number of trees at a certain number of feet apart. And then all the trees that will be planted will be Douglas fir. So then for the second diagram here, each of those little flags is a PVC pole, and that marks the center of each cluster. So for that top one, there's 4 trees with 4 feet in between And then we have all these different prescriptions, up to 36 trees at 10 feet apart. And then the clumping is opposed to the typical planting method that was mentioned of 350 trees per acre at consistent distances. So the thought process behind the different spacing between each tree and each clump is to figure out the optimal distance to maximize the understory growth, while also ensuring enough timber to sustain the root tree. So the use of gapping between the trees, as mentioned, allows for sunlight to penetrate through the canopy and provide the understory plants with a key building block for photosynthesis. So more and more studies have shown faults in the previous notion that nature, and specifically trees, is always in competition. And while sunlight, nutrients, and space are top limiting factors in many ecosystems, there's more connection and communication between trees than previously thought. So when considering this, I'm extra excited to discover the results of the study, as there are certainly not only one possible outcome. Considerations when designing the prescriptions and evaluating the results are that there may be large differences in the trees growing on the outside versus the inside of the clumps. As the edges are more susceptible to wind throw, which is, are they going to get knocked down? But they also receive more sunlight. So another consideration is that the different spacing between the Douglas fir will have varying impacts, like crown development, growth, or mortality. And as I was talking to Dr. Bobson about her expectations, I was interested to find out that the trees growing on the interior of a clump may grow taller faster because they compete more directly with nearby trees. And then another key thing to consider is what understory species will naturally regenerate in the gaps. And this can differ with gap size, proximity to the road, or closeness to the edge of tree clumps. And since we are not using herbicides on the majority of the sites, it will mirror very closely what would naturally occur. So how did we execute each prescription? Before we began, we would have to calibrate the head block, which is what Nate has in that picture over there. And we would calibrate it to 10 meters. And then we would use a georeference map to mark the boundaries between each plot with some flagging tape, so future crews can easily distinguish between the subunits. And then we use a plot map like this that is specific to each planting arrangement. And each dot here marks a PVC pole. With the closer together ones, we skip in between, but with the larger ones, we place a PVC in each dot there. And then one intern will stand at a previously placed PVC with a transponder or a birdie, which is on that pole. And then the other person will shoot the hadlock and get data on the distance and the azimuth, which is like the cardinal directions. And then if there's times when we're not sure what direction we should be shooting in, so we can use the north arrow here, and like if you're pointing north, the hadlock should display on 0 degrees, and so then you can move right or left to get it to the correct azimuth. And then for A and B blocks, we topped each PVC pole with a white paper plate to ensure LiDAR drone visibility when they create the maps. And then other tools we use, where we had our high-vis vests and then walkie-talkies. And whenever we finish a plot, someone says, "We finished the plot," and then we go, "Yay!" So that's pretty fun. Some troubleshooting. We use tablets with a program called Venza, and that is sometimes not the most accurate. So we did have to work around stuff like that. And then also the birdies, sometimes if they're too close to each other, you get interference. So at times we did think we had the right measurements, and then they might have been interfering, so we had to go back and redo it. So this summer is the final field season for pre-planting, data collection, and plot marking. And then in winter 2026, planting crews will implement trees according to the various prescriptions. And another consideration here is if the time to plant in clumps is the same or different from like planting in the place. And so how do they know where to plant the seedlings? PVC. Yeah, we definitely got well acquainted with PVC, putting in over 3,000 PVC pipes. And then at the end, we would have to mark each location with a post and a little tag, which were pretty hard to put in.
So like any experiment, we needed controls for T3 in order to understand how various prescriptions might be impacting tree growth. So as Anna mentioned, there were 4 blocks within T3 that we were working in, A, B, C, and D. Right here you can see C block, the yellow is a harvestable unit, and each of these black dots are plot centers, which we will get to in the next slide. But essentially all of these areas we visited were stands that are about 40 to 50 years old. They were almost all of Douglas fir. But usually we see something else, which was always exciting. And we were finishing up work from previous crews, and just going in and implementing permanent plots. So that future crews could go in and report data on the same trees, we could understand how they change over time. And relate this to other T3 sub-experiments, which focus on other stands of a similar age, which we didn't work on this year, but yeah, the experiments. So how do we do this? We took Avenza out. We drove out to the various different blocks, and we hike out through the forest. There was one time where we had like 3 miles or something crazy. And we would hike until we reached one of the plot centers, and then we would install a rebar and a PVC pipe in the center. And then we'd set up a jabot, which is that really accurate when the GPS device. And then we'd set up a transponder in the center of the plot. And next we would start measuring from the plot center. So any tree that was within a 10-meter radius of the plot, we would include. And the trees on the outer perimeter, we would spray paint with pink spray paint, so that we could locate things easy in the future. And the data we collected were species, EDH, which is diameter at breast height, which is 1.37 meters. We also collected data on LiDAR visibility, mortality, and any abnormalities that there might be. And another really crucial part of this was collecting the geospatial data from a tree to the center of the plot. And essentially we did this so that we can map these plots for the future, but also means that we might be able to do remote sensing on these plots, which would be very cool. And in these control plots, we added a Internship Douglas fir, a surplus of Pacific silver fir, a Western hemlock, the occasional red alder, and the rare Western red cedar. And Sophie was very insistent that I said these exact words. Okay, and the final part of T3 that we worked on this summer was the Western red cedar red alder polyculture experiment. Essentially, during the planning phase of T3, we heard from stakeholders and travel communities about concerns in declines of Western red cedar along the peninsula. They've been declining since the 1900s due to overharvesting and lack of replanting efforts, despite these trees being really culturally and ecologically important. So this concern gave rise to the polyculture experiment, which aims to learn how different planting ratios between Western red cedar and red alder would impact growth and stratigraphy, and also could have ecological impacts for other things. Yeah, because 2 trees grow together naturally, and as Nate mentioned, Western red cedar or red alder is nitrogen fixing. And it's also really fast growing species. Apparently, it's referred to as like weedy in a lot of timber operations, but it can be economically valuable for furniture building, and it can be harvested 35 years after planting, whereas Western red cedar takes about 70 years to be harvestable. So you can rotate these pretty well with the times that they're growing. And then the planting ratios are as follows: 100% Western red cedar and 0% red alder, 75% Western red cedar and 25% red alder, and then an even mix of the two, and then inversely 25% Western red cedar, 75% red alder, and then no Western red cedar and 100% red alder. And for this is another experiment that is in the last year of fruit planting. So we just went out to the various polyculture areas, and we flagged along the boundaries with different prescriptions so that winter when planting crews come in, we know we were planting up.
Surprise, we're also going to talk about soil processing. About years into the internship, I had an old player with chronic knee injury, so I ended up not being able to go to the field the rest of the time. But I got to take on the awesome project that is soil processing. So here at ONRC, there are over 100 samples of soil that was taken back in 2014 and 2015, and these were all taken from the outside site. What is the outside site? It is the long-term ecosystem productivity experiment. And it was created to test and compare various approaches to forest management. And there are 4 sites throughout Western Washington and Oregon, and the sites with the soil samples that I was working on were taken from Saco, Washington, which is about 10 miles away from Florence. And so the Saco site is pictured right on this top left corner. And all those different color rectangles are different units that are within each site. And each unit is given a very unique prescription. And this is a prescription combination between silviculture and woody debris, which are pictured in this table right here. And up top is the silviculture prescriptions. And so these prescriptions were put into place for each unit to kind of have a target sterile stage unit. So with the early sterile units, these are clear cuts that have been planted with 50% red alder and 50% Douglas fir. And the mid sterile units, these are also clear cuts that have been replanted with purely Douglas fir. In the late sterile units, these are a forest stand. They're about 50%. They're mostly hemlock and Douglas fir with a couple spruce mixed in, but these have been commercially thinned and not clear cut. And the control units are going to be roughly 90-year-old forest stands that have been completely untouched and unmanaged. And over here on the y-axis is woody debris. And so, as mentioned, with the early, mid, and late soil units, they've undergone some kind of management or harvesting or cutting. So with that, in logging operations, there's always going to be a little bit of woody debris left behind. So these percentages are specific percentages of woody debris that were supposed to be left behind just to test what effect woody debris is going to have on these stands. And so the number is assigned to each percentage. So 1 through 3, and as the numbers go up, that's the higher the percentage. And the only caveat to that is the control units because they have not been managed or touched at all. So there's no woody debris to be left behind. So those are the prescriptions of each unit. And next part is the component or the replication. So this study was built on a randomized block design. As you can see here, this is the same exact map with the same units, but they have been put into the blocks. And so this block design was based on spatial proximity because the terrain in Saco is pretty even, so there's no like weird elevation or anything, so it's just built based off of spatial proximity. And each of these units host the same distribution of treatments and the same number of units within each block. So each block has 3 early sterile units, 3 mid sterile units, 3 late sterile units, and 1 control unit. And the randomization part of this process is the location of each unit within each block. So those are all randomized. So that is a randomization element of how these blocks are put together. And so what is soil processing and what does that entail? So soil processing and soil sampling is a little different from a lot of other field data that is collected. Unlike things like measuring seedlings, observing tree heights, maybe like observing understory, that is all done in the field and then recorded right there in the field. The soil processing, it's a lot more work that is done outside of the field, which is why a lot of the soil samples that I processed were taken back in 2014 and 2015. So what I was doing with these samples is preparing them for nutrient evaluation, which is done at Windy River Hall at the Soil campus. And so the first part of this is getting rid of all anything larger than 4 milliliters, that's mostly roots and rocks, getting the weights on those, throwing away the rocks, saving the roots. And then you have like your fine sample of soil, and that is going to be run through the soil splitter, which pictured up top on the middle. And as you can see, there's slats throughout the splitter, which helps split the soil sample down in half, but it's also making sure that it is a representative sample of that larger one. So as you continue splitting it to be fit into that 4-ounce jar, which is pictured on the far left corner, it's going to still be a representative sample of that larger initial sample. So now you have your 4-ounce jar of fine soil, and it's going to be run through the soil grinder, which is pictured right on the bottom. And that's why you run it into a very, very fine powder with those 2 plates running opposite of each other with very, very high speed. You're going to get rid of very, very fine, dusty powder. Take that powder, goes back in the 4-ounce jar, and now it's ready for nutrient evaluation. And that is stored and sent off to the converter home. Yeah, so this summer I was able to process 192 soil samples, which was the entirety of the 2014-2015 LTEC samples that were taken. So it's very exciting. So you might be wondering, what are we going to do with this data? Why did we take all these soil samples? Why did I process soil samples that were 10 years old and 192 of them? So these plots have been monitored and measured on various indicators of forest health, such as tree heights, tree mortality, tree growth, understory retention. And so this kind of measurement and the indicators of forest health in conjunction with this variety of prescription just helps us make deeper connections with understanding forest health, and soil health is a great indicator of that. So these 2 graphs right here are both showing tree data, and I'm going to start with this pink graph on the left. So it's going to be a comparison of early sterile units as well as mid-sterile units, which are also known as negative Douglas fir, because they were the negative Douglas fir, but they're still mid-sterile units. On the x-axis here are the different blocks where each of the data was taken from, and on the y-axis here is going to be the biomass, which is the grams per hectare. And so as you can see, there is a very large discrepancy between B4 in the early sterile units and B4 in the standard Douglas fir units. So just that very large discrepancy. Continuing on with the basal area of each plot, the same discrepancy continues on the orange plot, where the B4 basal area is much higher than the B4 basal area of the mid-sterile units. So the key difference between these two units was the presence of red alder in the early sterile units. And as mentioned by my lovely colleagues, red alder is a fantastic nitrogen fixer in the soil. So it's really supplementing that soil with extra nitrogen, extra nutrients. So, however, it can't only be explained by the presence of red alder, or else all of these boxes and the other boxes would also be jumped up just as high, and there would still be that statistical significant difference. So because it's only block 4 that is showing this difference, there's definitely a presence of initial site-specific nutrients, or maybe some special conditions specifically at block 4. At block 4 that are just aiding the growth in some way. So this is just continuing, it's just helping us understand a lot more of the connection between red alder and site-specific soil treatments. And so on pre-testing of the site soil, there actually appeared to be higher levels of carbon, nitrogen, and the presence of a lot smaller rocks in block 4, which explains why those site-specific conditions are just a lot better for garden trees in general. So then add in the red alder, and it's just like an extra boost. It's like natural fertilizer. It's fantastic. And so these trends were taken in the 7th year of the LTEP study. And while these exact trends didn't continue after the 7th year, there were different trends that were observed. And year 20 of this experiment, which was when LiDAR data was collected on different tree heights. So this graph is showing block 1, 2, 3, and block 4 on the far left. And the blue box plot is representing the early sterile units, and the red box plot is the mid-sterile and the mid-sterile units. And as you can see again in block 4, there's a pattern between another discrepancy between the tree heights in early sterile units compared to mid-sterile units. And it's just continuing to show that over time, there is that connection between red alder and the presence of those site-specific conditions continue to have some residual effect on the trees that are growing there. And so that's just an example of like the kind of connections that you can draw between the site conditions and the presence of red alder, and how it all plays together, and how it just really affects tree height in general. It's just very interesting to watch in the long term if these patterns will continue, if maybe they'll diverge, if maybe they'll fell off. It's just a very interesting, very clever observing forest health, and it's really interesting to keep up with. Yeah. So.
During our time in internship, we found a lot of improvements in forests that we're very excited about. Not pictured here are the amount of berries we picked. Our hands were frequently purple by the end of the day from berries. We saw a lot of banana slugs, which by 3 of them being here and taking up a really hard slide, I'm sure you can tell we really love them. A lot of garter snakes, June bugs, a lot of frogs, but mostly at ONRC at night. We also saw one black bear. That was pretty fun.
So this summer, we also have the opportunity to go on some very exciting educational field trips. We went on 2. We are running a little short on time, so we won't be able to go as many of them as we would have hoped. But my highlight of the summer was when we were able to go on a field trip with Dr. Eric Trainer and Dr. Avery Hatters, who's we learned about who are with the Olympic Regional Harmful Algal Bloom Lab, ORHAB. So, we learned about what harmful algal blooms are, which are these massive growths of algae that can produce dangerous toxins, such as saxitoxin, that when it accumulates in shellfish can cause paralytic shellfish poisoning, which can cause serious illness and sometimes even death if contaminated shellfish are consumed. ORHAB was started back in 2000 when repeated harmful algal blooms caused closures of razor clam harvests along the Washington coast. And this resulted in big economic losses as well as public health concerns And it also affected tribal harvests. And so a few people, as a result, a few people came together, the tribal nations, the state agencies, and scientists like Dr. Trainer, and they formed or have in order to provide local monitoring and in faster warning systems and science that benefits both public health and all of us. And coastal economies. And today, ORHAB's monitoring acts as an early warning service. Seawater is regularly sampled. We actually got to help out with that one morning. And when harmful algal blooms are found, managers are able to close beach harvests before the shellfish become toxic, reducing of all of us being poisoned, which is great. So what stood out to me most about ORHAB is it's not just scientists in a lab, it is a true collaboration between tribes, coastal communities, the state, and NOAA scientists that are all working together to create new methods for testing, like the ELISA rapid test, which is similar in concept to the COVID rapid test, and takes 90 minutes, not several days. I can say a lot more, but these collaborations ensure that tribal sustenance and ceremonial harvests are protected along with commercial and recreational harvests. And it was really meaningful to be able to learn from the experts and to get a brief glimpse into what monitoring at this scale, how much of an impact it can have on tribal sovereignty and public.
So another highlight of the summer was our second field trip, where we went out to the Jimmy Lately experimental area on the east side of the peninsula with Dr. Bernard Norman. We checked out two pretty different forested stands. Both were pretty old, but each had a pretty different story to tell. On each of these stands, we set up a couple of these tree survey plots, very similar to the ones we did in T3 control units, to kind of see the difference in tree height and also forest structure, and hear a lot about the underlying story behind them. So the first unit was about 70 years old. It was first clear cut and then subsequently thinned about 30 years post replant. The effects of this thinning on the regeneration of both understory and also the arrival of new saplings, creating a more complex and layered canopy, was really prominent. It was really cool to see how this forest was almost managed to create older sterile habitat conditions similar to those that have been left untouched around the peninsula and in other places, with lots of complexity in the canopy and also on the forest floor. This creates very essential habitat for many endangered species around the peninsula, such as the marbled murrelet, which uses huge branches of the big trees to fly its eggs on and just leaves them there and hopes for the best, which is a good strategy. And we also, in contrast to that, visited another stand, which was much older, about 130 years, and that was cut, but just left to regenerate on its own. And those trees were very different. They were much smaller, much thinner, closer together. The canopy was in one single layer, and there was also pretty much no complete understory. This is what's known as a dog-eared unit, where there's many very skinny trees, almost like toothpicks, packed really close together. And these forests are really endangered, and they're hazardous for themselves because, well, they have very high competition for nutrients and sunlight because the canopy is so crowded. They're not able to grow a lot. They're not able to produce any defenses. to any pathogens or fetal outbreaks. And since there's really only one single structure in the forest, it's really susceptible to fire. Once one bit of fire gets in that canopy, the whole thing can light up and be gone. Even though the stand has looked like that for probably over 50 years now, it is pretty endangered. Another really interesting part of this story was the difference in ring growth that we saw between these two. You can see on the left side where Cello's standing on, shout out Cello. This was in the first unit where you can also see the data first on the graph. The planted stands initially had very wide rings, slowly tapering off as it went inwards. And then right after the commercial thinning, about 30 years after planting, there was a huge spike in the growth of these rings, indicating a burst of productivity in these individual trees. And what we saw in the dog hair stand was a much steeper taper. And also by the time the rings got to the edges, they were not visible to denote the differences at all. You would need a microscope or at least a very good hand lens to see that. But seeing different forest stands like this now bring up an interesting point about looking at forests in the past and how they were managed and applying lessons that you can learn about complex integrated management methods like commercial thinning, but also it highlights the importance of research projects like ethnoforestry and the T3 project, which aim at creating new and innovative approaches to forest management, hopefully creating viable habitat at all stages, early sterile, but also late sterile, because there's the same amount of species that depend on both around the peninsula.
Yeah, so we did have 3 weekends because our 40 hours was Monday to Thursday, so we got to spend a lot of our time camping, backpacking, playing games. We had to read all the Twilight books while we were in Forks. And then there were a few hardships. Our trucks did break down. We had to push it. But we got back and now we're family.
Thank you so much for coming and witnessing us talk about our summer. We had a really true blast at ONRC. I think everyone grew personally and also as scientists in forestry. And I think all the things that we're expecting to do in the summer. It's fantastic. A special huge thank you to Dr. Courtney Dobson and Dr. Bernard Gordon for just being fantastic mentors. And another huge shout out to our wonderful crew team for being with us. Thank you guys so much, and we're open for any questions.
Going back to the beginning, of taking the plant identification. Did you try any of these the apps that are advertised to identify plants, and also things like Google Lens? On your phone, just a picture of it.
They were useful to get things to like a family level, and then you could use like a family-based chapters and program it in to get a more specific ID. So like species level, no, but family level, they were sometimes helpful.
Yeah, things like seed they're wrong sometimes too, actually more often than not, you got it right, like maybe a couple times overall. And also
There's
there's,
what you pay for, but someone posted it free.
yeah, that's true. Yeah, having sparse service out there doesn't help either.
I had over 600 seed identification before the summer, and now I feel like that's all.
What's the future of this project gonna look like? Like, what's the time schedule?
Long time. Takes a long time.
Yeah, well, so this winter they're gonna be planting, and then post planting, it's a lot of like monitoring and coming back and seeing what the answers to these questions are. like we were talking about with like all the different restrictions, right? So like the understory plants and the gaps, and then like the differences between the interior exterior trees.
It would be maybe 50 to 170 years before these trees will actually be harvested, but hopefully we will be able to collect data in the meantime and get more meaningful data as time progresses.
One interesting thing, for any of these, but especially for things like, in the polyculture experiment, there's something, it's like late sterile designation for DNR land. So in, let's say 60 years, if something was designated as late sterile habitat, we have various for different DNR land. Blows down or catches fire, something like polyculture, one of our, experiments is going to be, logged, they're become, once they be late sterile habitat.
Maybe I'll jump in on that question, but first I want to thank all the interns and Nina for what was a very rigorous summer, and you guys did a huge amount of work, and your presentation was outstanding. You really described the work very, very well, and I want to thank you. Regarding how long these studies are going to go on, well, time will tell. It's not easy doing long-term research projects like these. The long-term productivity plots in Sappho have been running since, they've been running 30 years now, with a few more years before that where the monitoring was initiated. And so on the T3 study, we've been monitoring it for the last three or 4 years before treatments went in. Treatments are now in, and we will hopefully, I don't know how many will be around in 30 years to find out whether we succeeded in getting them. I think you interns will just be young people still, but it's not easy. It's really not easy to keep the funding coming to do this kind of work. But if our plan was to have an experiment that was so interesting, people just had to measure it.
I was wondering on the ethnoforestry and the understory, there are particular plants that you had been asked to find that you didn't find, what the diversity was within those plants.
So actually, Courtney, Dr. Bobson earlier had led a crew out to plant native species. She did not tell us to look for them.
Okay.
She was, I think, hoping that they were there. We did find a few, but I think not as many as she had hoped. And that, along with the fact that planting all of these different native seedlings is expensive, is one of the reasons that is not going to be implemented with scaled up versions of these plants.
I think that order was initially planted in that forestry, and it was a really big heat dome in the July that they were planted, and that caused a lot of them to get fried. So that was another element to take in, and some of the kind of like mixed funding and conservations.
I can chime in a little bit here as well. Yeah, so we have 5 different understory species that were planted. You're right, the crew did not measure them this summer, but they were measured for the first few years after implementation. However, the planting was done in winter of 2021 from mid to late February through early April based on plant availability, and we were in a pre-vaccine COVID world and had a lot of other barriers to getting a bunch of undergrads in the woods to plant things in close proximity to one another. But if you all remember 2021, that was also the year of the heat dome. And so we had a really weirdly warm April in Forks. I remember it being 80 degrees for a handful of days, followed by the June heat dome. So we had a lot of mortality of the understory species that were planted, unfortunately, which is in part why we have changed our monitoring approach to this. But yeah, we selected a handful of different species that would be useful for forage. It could be beneficial for local people to harvest and to try to think through some of these ideas of how do we take a small scale trial and potentially use some of these ideas in T3. And as the interns have said, I think we realized that planting understory species was probably not the best approach to T3. And so we're taking a different approach there.
What we also did was in the PVC class, we did understory class, which we made this little grid and we would identify the species in there. And with those, I would say that the species we found were pretty consistent. There was a lot of different blackberry and what was it? There's like oxalis and all of it. So yeah, so like some of these more like hardy plants that can grow in the heavy like wooded areas.
Did we want to talk more about the PVC and the paper plates? And what the paper plates are about?
We initially wanted to put the paper plates out so that when Miles, who's our like DNR counterpart, would fly a drone to take multi spectral and LiDAR imagery, I suppose. It would be easier to like I don't know how familiar people are with LiDAR, but it's essentially like a laser that sends out pulses and it gets returned to the drones and it can tell you like how far away something is from the ground based on how quickly those pulses return. So help us that with specifically multi-spectral, I believe, these plates would be really visible, so to be able to know exactly where the PVC were, and then it can help us for all the sensing stuff. To be determined how many of the plates stayed on the poles, we did fly drones, what, 2 weeks ago? And at least from those images, we cannot see the plates, but we found one. We found one. We also, the images need to be enhanced, so they might be there. Or be enhanced. Yeah.
More plates were found in the images afterwards, so don't worry. They can be located using the remote sensing data.
We have these like paper plates in our little high visibility vests in back pocket, and it'd be a whole ordeal. Sometimes we'd run out, we'd have to hike all the way back up to the top to get a new stack of paper plates. And we also unfortunately broke with Derek's drill bits at one point because we'd have to drill a hole in these plates and then put them on the stone. I believe it's under the Department of Corrections.
I've got one last question for you. After hiking up and down these slopes all summer, and you guys must have gone up and down, I don't know how many times. I'm wondering if any of you are going out for the UW crew tea.
Yes. This was a summer to get very in shape. It was also a summer of many falls.
So more spectacular than others.
They did like a full flip at one point.
Yeah. It's more of a twist.
Yeah, the terrain, the photos really don't depict how bad the terrain was, but I think that photo of me, when we first pulled up to T3 and saw how steep it was, kind of, yeah, we can zoom in.
But the best part is you can throw the PVC down the hill because it'll kind of roll and then you don't have to carry it all
the There's a Zoom question that asks, maybe this is a setup question, of what was the coolest wildlife you've seen this summer?
Oh,
Interpret coolest how you see fit.
So, on the first day out at ONRC, I was the first one to arrive, and I went up a little solo hike up the Reed Trail that is right next to ONRC, and it was a lovely hike. And I was coming back down on the gravel road, and up from the tree line comes a feline-shaped figure. So I take him out of the woods. I'm not really sure what it is, and I was like, you know what? It looks. I thought it was like a bobcat. I knew that was being. Oh, I couldn't see very well. It's down in the middle of the road. I'm like, I don't want to cross the path. Let me Courtney a call. Courtney, I'm describing this animal, and I go, yeah, it has like a long tail, and you know, oh, that's a cougar. That was a cougar that I ran into on the Reed Trail. It came back from the tree line. It was like 100-ish, 200-ish feet from it. It didn't see me. I'm still here to tell the tale. It was facing away from me, came down to the tree line, sniffed around, and then went back into the tree line and I ended up having to like still walk past that gravel and however past the tree line, but it was very adrenaline boosting. And I figured for the town. So I've seen a cougar in Pacific Northwest. I think it's great.
It also sparked cougar mania afterwards. You're all for like 2 weeks. We hear a noise. We're like, oh my god, it's
But no, I did.
and I saw glowing eyes in the woods right after hearing the noise. So I screamed.
Oh,
How do you break the helmet with the paper plate?
Big stack, like
Yeah.
they're trying to get through the.
Yeah.
I think what happened, it was on two metal beams and I think I got far enough down that it just.
Alex, what have there another question?
Yeah.
Oh, go ahead, go for it.
Okay, is there any? Did y'all see any, or is there any medical correlation between like what the fire regime might be out here? And how much like carbon gets captured in the soil? Anything like that that might help these plots and or not help these plots if a fire comes through or they burn piles and they.
Yeah, but I would like to talk about the soil side of that, but at least for polyculture, something we learned about on our field trip with Bernard was apparently when you plant red alder, it can like really change up how fire can travel through the forest because it there's just like so much nitrogen in alders and it has a like higher. It's like a I don't know, like it has a higher water content or something like that and adds more organic matter to the soil. So essentially it can just kind of like, it takes more energy for the fire to burn through those areas that have alder. And there was a grad student, Nick, who came out on that field trip and he has this idea of doing like rings of gaps between trees and planting alder, and then that also could help with slow fires that it goes through. So yeah, that's kind of implemented through T3.
We also learned on a more basic level that patches of trees at different heights can slow the fire.
On the soil stuff, from what I remember, when we went out on the field trip with Dr. Gorman, we did end up seeing soil pit that had some charcoal that was really deep down. I forgot which soil layer it was specifically, but a lot of that like charred material kind of up very deep into the soil, which can then render the soil hydrophobic, which could be a big barrier to like regeneration and just like, especially like the clear cuts and wasn't thin, it's just kind of a big like it's a big block to like try to actually regenerate. Yeah, and with like planting the clear cuts, since all the trees are clear at the same time, they all end up like relatively the same height. So with the different heights of the trees, like it'll just kind of go through all your ground.
Yeah, it very much depends on the structure of the forest. I mean, most of the forests we were working at were complete clear cuts with lots of slash. So there's not a lot of nutrient cycling, active nutrient cycling going on anyway. Lots of fuel. Yeah, lots of fuel. But like getting those more complex layered forests with alder and different canopies. Like there could be a more trickly based fire that actually helps ignite the duff and release nutrients into the soil if it's not super deep and doesn't just like a more severe fire would just incinerate that whole liquid. But fire on a smaller, less severe scale can actually help cycle those nutrients.
In any of these clear cuts, had there been the debris piles burned? And if so, was there any noticeable difference in how the trees, planting trees, responded?
We saw many piles, they did not appear to be burned.
They're not, they don't seem to burn.
There was some burn stuff, but it seemed like it was probably intentional, like there were a lot of like really big, like huge stumps that had been charred for whatever reason, but it didn't seem like it changed, like any of the understory that was around.
We have a question on Zoom if you're ready. The question is, what did you enjoy most about the internship and what is the biggest takeaway of your experience?
Yeah, so I'm sorry. I really enjoyed learning plant ID at the beginning of the summer because as I mentioned previously, I'm a seed enthusiast. And when I discovered that it has been lying to me for all these years, I was very excited to learn how to actually ID plants for the first time. And I really grew in my capacity and I feel like there were two parts. My favorite experiences have also been exploring the tiger cluster. So incredibly cool. And all of the tide pools, sea stars.
I think for me, like my favorite work-related thing was when you'd be measuring between the posts and you like perfectly line things up on the first try. It's very satisfying. And then out of work, just like meeting all of these wonderful people and having chats with them and being able to explore the peninsula and learn all these wonderful rivers and beaches and hikes and yeah, just a very lovely community here and taking a lot of new friends into the next year.
Yeah, no, I agree. I think, yeah, with plot implementation, it's long and sometimes takes multiple days of steep climbing over slash and falling down these hills to complete a plot. So when you actually do complete one, it's very satisfying. And along with that, it's very interesting to see how projects like these actually do get implemented on these large scales. A lot of times there's like a disconnect when you're learning about these things in class and reading the data set, and you don't really, especially in natural sciences and forestry, there's a lot of groundwork that needs to be done. And it was great to be part of that. And my takeaway, yeah, the coast, the whole peninsula, the forests, so many cool rocks and islands and waves to discover.
When I was in the field for the first 2 weeks, like the most rewarding thing to me, I'm not from Washington, I'm from Virginia, and so I was identifying these native plants. And so it started to go to the point where I would start to get them right on the first try. So I got to check that off. That was very, very fulfilling. And then when I was working on the soil, I think the most fulfilling part is when I finally finished, I finished yesterday. And I looked back at like the warehouse and everything was really neat and ordered me, and I was like, whoa, it's like all been processed and like by me, like that was just really, really rewarding for them. And then personal favorite part of the trip is getting to know everyone as well, and just kind of going out on adventures around the peninsula, playing games together. It's just like a very good like bonding relationship.
Yeah, work related. I really enjoyed the field trip with Dr. London. I thought it was really interesting that one of the courses we looked at could have been like health return intervention and like within, because typically I would think that like it's best to leave nature alone and let it, you know, take its course. But and like hearing that it could have actually improved the health of the course was pretty interesting. And then non-work related, I really like learning.
Who's coming back?
Well, if you'll have us, yeah. Any more questions? Yeah, questions? We wish. I really want to. I was looking to do that in soil processing, but actually talked to Dr. Towers about this apparently because the soil is very old. There's like no way to see those live. Microorganism communities, and so I think best to do like fresh and feel like immediate disaster. Yeah, I really want to though.
Courtney, do we have any more Zoom questions or?
No, I don't think so. Yeah, really wonderful presentation, everybody. Yeah, thank you so much. And thanks to everyone who joined in person and on Zoom.
Thank you all.
Thank you everybody for coming out. It's been quite a pleasure to share everything with you.